Hello everyone and welcome back to my channel. So for viewers who are new to my channel, I'll just give a brief introduction. So I am Triya and I generally post lectures and videos on tissue engineering, regenerative medicine and its associated related topics. So in my previous classes, I have discussed about the basics of tissue engineering and how the important integral parts of tissue engineering are important. For example, the scaffold fabrication techniques. Now, I thought that before going details into the tissue engineering, it becomes important for us to understand the basics about the ECM, the cell, the biology behind it, so that we can have a better understanding. So before solving any problem, it becomes important to understand the biology of it. So for that, I have started the lecture which is on the cell and the ECM interactions, which will be in two parts. So this is the part one. So let's get started with this today's lecture. We'll first start with understanding the general organization of tissues. So in order to understand the interactions that takes place between the cell and the extracellular matrix, it becomes important for us to understand what the generally the organization of tissues is. So as it is visible in the chart, so there are majorly four types, which is the nervous, the epithelium, which is also known as the parenchyma, the muscle type, and the connective tissue type. So talking about the first one, which is the nervous type, so we'll discuss about the embryonic origin of it, like from where it has origined, the major tissues where it is found, and the cell types. So the embryonic layer from where nervous type is originated is the ectoderm, which is also known as neuroectoderm. The major tissues are the central nervous system, the retina, the peripheral nervous system, which is written as PNS, and the pigment cells. Further, discussing about the cell types, which are the neurons, the glia cells or the glia cells, the melanocytes, and others. Talking about the second type, which is the epithelium or parenchyma. So it has three different embryonic layer origins. So it can be either ectoderm, like in tissues, skin, hair, and mammary glands. And the cell types include the keratinocyte and others. Coming to the endoderm, so from endoderm, tissues like gut, liver, pancreas, lung, and other glandular organs have originated. And their cell types are various and numerous cell types. For example, only in liver there are 6 and 7 types of cells which are being present. Talking about the mesoderm, so from mesoderm, urogenital tract, mesothelium and endothelium has originated, and cell types include the mesothelial cells and the endothelial cells. Further, talking about the muscle cell type or the muscle type, it has originated from mesoderm, and the major tissues include the skeletal, the cardiac and the smooth muscle. And the major cell types include the myocytes, which are the tubular cells, and the smooth muscle cells, which is also known as SMC. Talking about the fourth type, which is the connective tissue type, it has majorly originated from mesoderm and it is found in the interstitium and in blood. The major cell types include the fibroblast, the red blood cells, neutrophils or polymorphoneutrophils, then monocytes, also macrophages, then other innate immune system type cell types like the eosinophils, the basophils, lymphocytes, platelets, and multiple such precursors. Now discussing about why there is need and why do all the multicellular animals have extracellular matrix. So tissues are not made up of solely of cells. A substantial part of their volume is extracellular space, which is largely filled by an intricate network of macromolecules which constitutes the extracellular matrix. This matrix is composed of a variety of proteins and polysaccharides that are secreted locally and assembled into an organized meshwork in close association with the surface of the cell that produced them. So, we have discussed in our previous lectures about the cell junctions that are chiefly in context of the epithelial tissues. Our account of the extracellular matrix concentrates on the different types of tissue which we have discussed in the previous slide. So coming to why there is need for multicellular animals to have the ECM, which is divided into 4 parts, which is it acts as structural support to maintain cell organization and integrity in the epithelial tubes, the mucosal lining of gut, the skeletal muscle and fiber integrity. It also provides the hardness to bone and teeth. For example, collagen fibrils become mineralized. Further, it presents the information to the adjacent cells. For example, inherent signals like the RGT motifs in fibronectin, which helps to bind to the integrant molecule of the cell and helps in the cellular attachment, and others like the bound signals, which includes the BMP7, TGF-beta, FGF and SHH. Further, it also serves as the highway for cell migration during developmental, like the neural crest migration, in normal tissue maintenance, like in the intestinal mucosa, and also in the injury or in the disease, for example, in the wound healing and cancer. Further, we'll be discussing about the different types of ECM components. So starting with the basics for everyone to understand. In biology, the extracellular matrix is a three-dimensional network that consists of extracellular macromolecules and minerals like collagen, enzymes, glycoproteins, and hydroxyapatite that provides structural and biochemical support to the surrounding cells. Because multicellularity evolved independently in different multicellular lineages, the composition of ECM varies between the multicellular organisms and multicellular structures. However, the cell adhesion, cell-to-cell communication, and differentiation are common functions of the extracellular matrix. So the animal extracellular matrix, it includes the interstitial matrix and the basement membrane. So interstitial matrix is present between various animal cells, that is in the intercellular spaces, and something like gels of polysaccharides and fibrous proteins, they fill the interstitial space and act as a compression buffer against the stress that is placed on the ECM. Further, coming to the types of ECM components, so as I already mentioned, it consists of fibrous structural protein like collagen and elastin, and proteoglycans like hyaluronic acid, and large adhesive glycoproteins like the integrins, laminins, fibronectin, vitronectin, thrombospondins, and tenascins. So the basement membrane, as I already mentioned, is like a sheet-like depositions of ECM on which various epithelial cells they rest. Each type of connective tissue in animals has a type of ECM, like the collagen fibers and bone mineral comprise the ECM of bone tissue, and reticular fibers and ground substance comprise the ECM of loose connective tissue, and blood plasma is the ECM of blood. Further, there are several components of ECM. So one can imagine ECM as a hydrated network that comprises of various types of molecules that I already mentioned in the previous slide. One of the important molecule is the collagen, which is the most ubiquitous structural protein, and it is characterized as a triple helical protein that contains peptide chains with repeating of glycine and X and Y, which is usually the proline triplets, and the triple helix that is formed with the association of the three related polypeptides, which is the alpha chains. They form a coiled coil with the side chain of every third residue directed towards the center of a superhelix. And there is also steric constraints that dictate the center of the helix to be occupied only by glycine residues. Further, many proline and lysine residues are enzymatically converted to hydroxyproline and hydroxylysine, and around 28 distinct collagen types, each is assigned a Roman numeral that generally delineates the chronological order in which the collagens were isolated or were characterized. Coming to the collagen triple helix or type 2 helix, it is a primary secondary structure of various types of fibrous collagen, which includes the type 1 collagen. It consists of a triple helix that is made up of repeatase amino acid sequence, as I already mentioned, containing the sequence of glycine X and Y, where X and Y are frequently proline and or hydroxyproline. So collagen is generally folded into a triple helix, which is also known as tropocollagen, and collagen triple helices are often bundled into fibrils, which themselves form the larger fibers, as in tendon. Further, the glycine, proline, and the hydroxyproline must be in their designated positions with the correct configuration. More details I'll be giving in the future and the further slides. This slide shows the diversification of the collagens, which ranges from type 1 to type 16 and 17. This chart gives the overview of the various types and how it is like where it is found and in which form it is found there. So giving some examples, which is for example type 1, which is the found in as fibrils, and it is being found in the skin, tendon, bone, ligaments, dentin, and interstitium. Similarly, type 2 is also found in the form of fibrils, and it is found in the cartilage and the vitreous humor. Further, the type 3, it is also found in the form of fibrils, and it is found in skin and muscle. Similarly, type 4 is found as 2D sheets in all the basement membranes, and type 5 and 6 are found with like fibrils with globular end, and it is found respectively in cornea, teeth, bone, and also in some interstitial tissues. Now we'll move on to discuss about details of different types of collagens. So starting with the fibrillar collagens, which is the 1, 2, 3, and 5. So the fibrillar collagens, they constitute a subgroup within the collagen family, of which there are 28 types in humans, whose functions are to provide three-dimensional frameworks for tissues and organs. These networks confer mechanical strength as well as signaling and organizing functions through binding to cellular receptors and other components of the extracellular matrix. Here we'll describe about the structure and assembly of fibrillar collagens and their procollagen precursors from the molecular to the tissue level. The structure of the collagen triple helix is influenced by the amino acid sequence, the hydrogen bonding, and the post-translational modifications such as prolyl 4-hydroxylation. There are numerous steps in the biosynthesis of the fibrillar collagens, and they are reviewed with particular attention to the role of the prolyl 3-hydroxylation, collagen chaperones, trimerization of procollagen chains, and proteolytic maturation. The multiple steps controlling fibril assembly are then discussed with a focus on the cellular control of this process in vivo. Our current understanding of the molecular packing in collagen fibrils from different tissues is summarized on the basis of various experiments. And further, we'll go into details about the structure and how it is formed. Further going deep into the functions and where it is found, I would like to mention that among the collagens, fibrillar collagens are present from sponges to humans, and they are involved in the formation of the well-known striated fibrils. So there are different steps in the evolution of this protein family, from the formation of an ancestor fibrillar collagen gene to the formation of different clades. Certain genomic data from the guanoflagellate and from diploblast animals certainly have suggested that there are formation of an ancestral alpha chain that occurred before the metazoan radiation. And certain phylogenetic studies have suggested an early emergence of the three clades that were first described in mammals. Hence, the duplication events leads to the formation of the A, B, and C clades occurred before the eumetazoan radiation. So this is some historical and evolutionary details. Further, going into back into the fibrillar collagens. So the connective tissue proteins, they provide the tensile strength, and triple helix, as already mentioned, they are considered of 3 alpha chains, and glycine is present at every third position, and it is in the format of glycine X and Y, where X and Y can be proline or hydroxyproline. It is very rich in the proline content, and there is requirement and need for hydroxylation for proper folding. It is generally found in bone, skin, tendons, cartilage, and arteries. Now we'll discuss about bone. So bone is a natural composite of collagen, protein, and the mineral hydroxyapatite. The structure of bone is known to be the important to its load-bearing characteristics, but it is relatively little known about the structure or the mechanism that govern the deformation at the molecular scale. So here we'll discuss about it. So bone is like 70% mineral and 30% protein, and mostly collagen. So certain mutations of collagen genes can lead to a wide range of disease, from mild wrinkling to brittle bones to fatal blistering of the skin. So it has been found in several researches and studies that as the mineral density increases, the tensile modulus of the network also increases monotonically and well beyond that of pure collagen fibrils. Certain studies suggest that the mineral crystals within this network bears up 4 times the stress of the collagen fibrils, whereas the collagen is predominantly responsible for the material's deformation response. Findings suggest that the mechanism by which bone is able to achieve superior energy dissipation and fracture resistance characteristics beyond its individual by constituents. The two major constituents of bone, which is the soft collagen protein and the much stiffer apatite material. So during the formation of bone, collagen molecules assemble into fibrils, which are mineralized via the formation of apatite crystals. Although larger scale bone structures differentiate depending on the bone type and species, the structure of mineralized fibrils is highly conserved across species and different types of bone, and hence they act as bone's universal elementary building block. The hierarchical structure of bone enables it to be lightweight material that can carry large loads and combines the toughness of inorganic material and flexibility of protein-based tissues. Certain previous studies and research showed that the mechanical models have uncovered the key mechanistic features of bone as well as eluted the role of mineral platelets in strengthening the material. Further talking about the matrix assembly and how it is formed. So in the process of matrix assembly, multivalent extracellular matrix proteins are induced to self-associate and to interact with the other ECM proteins to form fibrillar networks. Matrix assembly is usually initiated by ECM glycoproteins that binds to the cell surface receptors, such as fibronectin dimers binding to the alpha 5 beta 1 integrin. Receptor binding stimulates the FN self-association, which is mediated by the N-terminal assembly domain and organizes the actin cytoskeleton to promote cell contractility. FN conformational changes expose additional binding sites that can participate in the fibril formation and in conversion of fibrils into a stabilized insoluble form. Once assembled, the FN matrix impacts the tissue organization by contributing to the assembly of the other ECM proteins. So there are several steps, molecular interactions, and cellular mechanisms involved in assembling the FN dimers into fibrillar matrix, while there are also important issues and major questions that requires further research and investigation. So the principal function of the extracellular matrix protein, fibronectin, is to connect cells to the matrix that contain fibrillar collagen. At least 20 different forms of fibronectin have been identified as of yet, and all of them arise from alternate splicing of a single fibronectin gene. The soluble forms of fibronectin are found in tissue fluids, and the insoluble forms are organized into fibers in the extracellular matrix. Further, fibrin, heparin, sulfate, proteoglycan, and collagen, they bind to distinct regions in fibronectin, and they integrate fibronectin fibers into the extracellular matrix network. Further, some cells express integrin receptors that binding to the arginine, glycine, and aspartate, which is also known as RGD sequence of fibronectin. Further, elastin is a key extracellular matrix protein that provides resilience and elasticity to tissues and organs. Elastin is roughly 1,000 times more flexible than collagen. Thus, the main function of elastin is the elasticity of tissues, and hence it got its name. So as I already mentioned, that the principal function of elastin is to impart elasticity to tissues, and elastin monomers, which is known as the tropoelastin subunits, are organized into fibers, and the fibers are so strong and stable that they can last a lifetime. The strength of elastic fibers arises from covalent crosslinks that is formed between the lysine side chains in adjacent elastin monomers. Further, the elasticity of elastic fibers arises from the hydrophobic regions, which is stretched out by tensile forces, and they spontaneously reaggregate when the force is released. So elastin, like collagen, is a protein which is a major constituent of ECM, of connective tissue. It is basically present as a long and flexible strands in soft tissue. In contrast to collagen, elastin fibers do not exhibit a pronounced hierarchical organization, and elastin is linearly elastic material that changes the deformation and has very small relaxation effects. Laminins are a major component of basal laminin, and this protein can occur in different forms, which results from the closely related gene translation. So laminin shows a high binding affinity for cell surface as well as for heparin and collagen type 4. Cell adhesion results from the presence of specific binding motifs that includes the RGD, which is the arginine, glycine, and aspartate, and PTHGR, which is proline, aspartate, serine, glycine, and arginine, and they can interact with cell-specific surface receptors. Because of these binding capabilities, laminin is often applied to coat cell culture dishes and tissue plates to facilitate cell attachment and spreading. Further, laminin is a complex adhesion protein that is found in the extracellular matrix, especially within the basement membrane. And this protein also plays an important role in early embryonic development and is perhaps the best studied of the ECM proteins found within embryonic bodies. Laminin is a trimeric cross-linked polypeptide that exists in numerous forms dependent upon the particular mixture of peptide chains. Laminins are a family of extracellular matrix protein, and as already mentioned, they are found in virtually all tissues of vertebrate and invertebrate animals. The principal functions of laminins are to provide an adhesive substrate for cells and to resist the tensile forces in tissues. Laminins are heterotrimers that comprises three different subunits wrapped together in a coiled-coil configuration, and laminin heterotrimers do not form fibers. They basically bind to the linker proteins that enable them to form complex webs in the extracellular matrix. Further, a large number of proteins they bind to laminins, including more than 20 different cell surface receptors. Vitronectin is a glycoprotein of the hemopexin family, which is abundantly found in serum, extracellular matrix, and in case of bones, it is also found. Vitronectin generally binds to the integrin alpha 5 beta 3 and thus promotes cell adhesion and spreading. It also inhibits the membrane damaging effect of the terminal cytolytic complement pathway and binds to several serpins, which is serine protease inhibitors. It is a secreted protein and exists in either a single chain form or a clipped two-chain form held together by a disulfide bond. Vitronectin has been speculated to be involved in hemostasis and also in tumor malignancy. Talking about its structure, It is a 54 kilodalton glycoprotein that consists of 478 amino acid sequences. About one-third of the protein's molecular mass is composed of carbohydrates, and on occasion the protein is cleaved after arginine 379 to produce two-chain petronectin, where the two parts are linked by a disulfide bond. No high-resolution structure has been determined experimentally yet, except for the N-terminal domain. Further, the protein consists of three main domains: the N-terminal somatodenine B domain, which is 1 to 39, and the central domain with hemopexin homology, and a C-terminal domain also with hemopexin homology. So as already mentioned, it is an extracellular matrix protein and it circulates in blood plasma in its soluble form. Further, vitronectin can bind to many different types of proteins such as collagens, integrins, clotting factors, cell lysis factors, extracellular proteases, and it also facilitates the targeted cell adhesion during blood clotting. Further, vitronectin also facilitates blood clot formation in damaged tissues. In order to target deposition of clotting factors in tissues, vitronectin must convert from the soluble form to the insoluble form, which binds the clotting factors. The major biological function of proteoglycans derives from the physicochemical characteristics of the glycosaminoglycan component of the molecule that provides the hydration and the swelling pressure to the tissue, which enables it to withstand compressional forces. So the proteoglycans are basically the glycosylated proteins which have covalently attached highly anionic glycosaminoglycans, and many forms of the proteoglycans are present in virtually all extracellular matrices of connective tissues. The major biological function or physicochemical characteristics of the glycosaminoglycan component of the molecule, which provides hydration and swelling pressure to the tissue, enables it to undergo and withstand compressional forces. The function is best illustrated by the most abundant proteoglycan in cartilage tissue, which is the aggregan. During the past decade, diverse species of proteoglycans have been identified in many connective tissues, on cell surfaces and in intracellular compartments. These proteoglycans have distinct biological functions apart from their hydrodynamic functions, and their involvement in many aspects of cell and tissue activities has been demonstrated. So the proteoglycans consist of a central protein core to which long linear chains of disaccharides called the GAGs are attached, and the GAG chains on proteoglycans are negatively charged. This gives the proteoglycans a rod-like bristly shape due to charge repulsion. Proteoglycans consist of a central protein core to which long linear chains of disaccharides GAGs are attached, as already mentioned, and the GAG bristles act as filters to limit the diffusion of viruses and bacteria in tissues. Proteoglycans, they attract the water to form gels that keeps the cell hydrated, and they cushion tissues against hydrostatic pressure. They basically bind to the ECM, other components like the structural proteins and other cell surface receptors. Now, after understanding how the network is built up of the extracellular matrix and how it maintains its integrity, it becomes important for us to understand that the ECM is constantly degraded and then remodeling is occurring. So proteases function in degrading the ECM components. So the cells must routinely degrade and replace their ECM as a normal part of development and wound healing. So the ECM proteins are degraded by specific proteases which cells secrete in an inactive form. These proteases are only activated in the tissues where they are needed. Activation usually occurs by the proteolytic cleavage of a propeptide on the protease. The matrix metalloproteinase, which is the MMP family, is one of the most abundant class of these proteases. It can degrade all of the major classes of extracellular matrix proteins. Further, the matrix metalloproteinases, which is also known as MMPs, can activate one another by cleaving off cleaving of their propeptides. So, therefore, it results in a cascade-like effect of protease activation that can lead to rapid degradation of extracellular matrix proteins. Further, talking about the integrins. So most of the integrins are receptors for the ECM proteins. So virtually all animal cells, they express integrins, and they are the most abundant and widely expressed class of extracellular matrix protein receptors, and some integrins they associate with the other transmembrane proteins. Integrins are basically composed of two distinct subunits, which are known as the alpha and the beta chains, and the extracellular portions of both the chains bind to the extracellular matrix proteins. The cytoplasmic portions they tend to bind to the cytoskeletal and signaling proteins, and in vertebrates there are many alpha and beta integrin subunits which combine to form at least 24 different alpha beta heterodimeric receptors. Most cells they tend to express more than one type of integrin receptor, and the types of receptor expressed by a cell can change over time or in response to different environmental conditions. Continuing to the topic, so integrin receptors bind to a specific amino acid sequence in a variety of extracellular matrix proteins, and all of the known sequences contain at least one acidic amino acid, and the integrins are also the signaling receptors that control both cell binding to extracellular matrix proteins and the intracellular responses that follows the adhesion. Integrins have no enzymatic activity of their own. Instead, they tend to interact with the adapter proteins that link them to the signaling molecules. Talking more about the ECM or the extracellular matrix. So the glycosaminoglycans or the GAGs, they are covalently linked to protein and the proteoglycans forming gels. So the fibrous proteins which includes the collagen, elastin, fibronectin and laminin. So there is repeating disaccharide sequence of a GAG sulfate. Consequently, there are carboxyl groups and negative charges. Different types are the components are the hyaluronan, the chondroitin sulfate, dermatan sulfate, heparan sulfate, and keratin sulfate. As you can see in this image that there is repeating disaccharide of the hydronic acid and N-acetyl galactosamine 4 sulfate. This slide shows some of the common proteoglycans and what are the major functions of it. So talking about the major functions of proteoglycans, so they constrain the range of embedded protein actions and affect the embedded protein activities. They also tend to control embedded protein release rate and prevent the embedded protein degradation. It also can function as co-receptors. Talking about some of the most common proteoglycans, which include the aggrecan, beta-glycan, decortin, perlican, syntican, and DALI, which is found only in Drosophila. So this chart gives an idea about the molecular weight, the type of GAG chain. So it is important for us to understand that there are different GAG chains that assembles to give result to the respective proteoglycan. For example, in case of aggrecan, it is composed of the chondroitin sulfate and keratin sulfate, and the number of GAG chains are around 130. It is majorly located in cartilage and it functions as to support in form of mechanical support, and it forms large aggregates with hyaluronan. Consequently, beta-glycan is composed of the chondroitin sulfate, dermatan sulfate comprising of one chain, and it is found in the cell surface and in the matrix. It basically functions to bind to the TGF-beta. Another important proteoglycan, which is the decorin, it is composed of chondroitin sulfate and dermatan sulfate of one GAG chain, and it is widespread in the connective tissue. It basically binds to the type 1 collagen fibrils and to the TGF-beta. Further, the perlican, which is composed of heparan sulfate chains of range from 2 to 15 and found in basal lamina, it functions as the structural and the filtering function in the basal lamina. The other two proteoglycans, which is the syndican and the DALI, they also are found in different locations of epithelial cell surface and cell surface respectively, and which functions in cell adhesion by binding to FGF and other growth factors. And in case of DALI, which is found only in Drosophila, it acts as the co-receptor for wingless and the decapitangelic signaling protein. Further, now we'll discuss more details about the fibrous proteins of the collagen, elastin, fibronectin, and laminin. As already mentioned briefly about the collagen molecules, which comprises about the glycine, proline, and hydroxyproline, and it is basically intertwined, like three fibers are intertwined together in order to form triple helix. It is mostly found during the embryonic chick's skin development, and the fibroblasts are surrounded by the collagen fibrils. The basic formation of collagen crosslinks is attributable to the presence of two aldehyde containing amino acids, which basically react with each other amino acids in collagen to generate difunctional, trifunctional, and tetrafunctional crosslinks. A necessary prerequisite for the development of these crosslinks is that the collagen molecules be assembled in the naturally occurring fibrous polymer. And once this condition is met, cross-linking occurs in a spontaneous progressive fashion. The chemical structures of the crosslinks dictate that very precise intermolecular alignments that must occur in the collagen polymer. And this seems to be a function of each specific collagen because the relative abundance of the different crosslinks varies markedly depending upon the tissue of origin of the collagen. So it is basically deaminated by the lysyl oxidase to yield aldehyde groups, which is spontaneous covalent bond between the aldehyde groups every like about every 67 nanometer, and if the cross link is broken, collagen is easy to tear. Collagen fibril formation is basically a self-assembly process, that is one which is to a large extent determined by the intrinsic properties of the collagen molecules themselves. But it is also sensitive to cell-mediated regulation, particularly in young or healing tissues. Recent attention has been focused on the early fibrils or the fibril segments of approximately 10 microns in length, which appear to be intermediates in the formation of mature fibrils that can grow to the length or length of hundreds of micrometers in length. Data from several laboratories indicate that these early fibrils can be unipolar, which means that with all the molecules pointing in the same direction, or either bipolar, in which the orientation of the collagen molecules reverses at a single location along the fibril. The occurrence of such fibrils, early fibrils, has major implications for tissue morphogenesis and repair. So, as it is visible that the collagen fibrils are basically synthesized and released, starting with the synthesis of the pro-alpha chain, followed by hydroxylation of selected prolines and lysins, which is then followed by the glycosylation of the selected hydroxylysins. Further, there is self-assembly of the three pro-alpha chains, followed by pro-collagen triple helix formation and the secretion of the synthesized collagen. Continuing with the discussion, so after the secretion of the pro-collagen molecule, there is cleavage of the propeptide. So I've already mentioned once in the previous one of the slides that the pro-collagen molecule or the pro molecule is cleaved and then that results into release or formation of the collagen molecule. Further, there is self-assembly of these into fibril form of around 10 to 300 nanometers, which further the collagen fibrils, they form the collagen fibers. And the collagen fibers range in the diameter of 0.5 to 3 micrometer, and there is also aggregation of collagen fibrils to form the collagen fiber. This slide shows the elastic fibers that consist of elastin, which I already mentioned are basically they function for maintaining the tissue elasticity and for providing the tissue elasticity. And this is actually the electron micrograph image of the dog iota, where we can see that the elastic fibers are so beautifully seen, and we can appreciate that how they are distributed all over the place in order to provide the tissue the ideal elasticity. Coming to the elastic fibers, so stretching of a network of elastin molecules. So elastin is highly hydrophobic and there are mainly two features, the hydrophobic and the cross-linked segments. The hydrophobic segments, they are responsible for providing the elasticity and the cross-linking provides the stability. So the elastic fibers are an essential component of the extracellular matrix composed of bundles of proteins which are known as elastin, which are produced by a number of different cell types including fibroblast, endothelial cells, smooth muscle cell and airway epithelial cells. These fibers are able to stretch many times their length and snap back to their original length when relaxed without loss of energy. Elastic fibers, they include the elastin, elaunin and the oxytocin. So elastic fiber is classified as connective tissue proper and elastic fibers are formed via elastogenesis, which is a highly complex process that involves several key proteins including fibulin 4, fibulin 5, latent transforming growth factor beta, binding protein 4, and microfibril associated protein 4. In this process, tropoelastin, the soluble monomeric precursor to elastic fibers, is produced by elastogenic cells and chaperoned to the cell surface. Following excretion from the cell, the tropoelastin self-associates into 200 nanometer particles by coacervation, which is an entropically driven process involving interactions between the tropoelastin's hydrophobic domains, which is mediated by glycosaminoglycans, heparin, and other molecules. These particles then fuse to give rise to 1 to 2 micron spherules, which continue to grow as they move down from the cell surface before being deposited into the fibrillin microfibrillar scaffolds. So, following the deposition into microfibrils, tropoelastin is insolubilized via extensive cross-linking by members of the lysyl oxidase and lysyl oxidase-like family of copper-dependent amine oxidases into amorphous elastin, which is a highly resilient insoluble polymer that is metabolically stable over a human lifespan. There are two families of enzymes that react with many lysine residues present in the tropoelastin to form reactive aldehydes and L-lysine via oxidative deamination. These reactive aldehydes and L-lysines can react with one another lysine and L-lysine residues to form desmosine, isodesmosine, and a number of other polyfunctional crosslinks that join surrounding molecules of tropoelastin into an extensively cross-linked elastic matrix. This process thus creates a diverse array of intramolecular and intermolecular crosslinks. These unique crosslinks are responsible for elastin's durability and persistence. Further, maintenance of the cross-linked elastin is carried out by a number of proteins including lysyl oxidase-like 1 protein. Mature elastic fibers tend to consist of an amorphous elastin core surrounded by glycosaminoglycans, heparan sulfate, and a number of other proteins such as microfibrillar associated glycoproteins, fibrillin, fibulin, and the elastin receptor. Coming to another important protein, which is fibronectin. So fibronectin is a critically important ECM protein that helps to mediate the cell to ECM interaction during fundamental events such as development, wound healing, fibrosis, and tumor progression. Fibronectin is secreted as a soluble, covalently bound dimer of 440 kilodalton molecular weight, and fibronectin is encoded by a single gene. However, different forms of fibronectin arise through alternative splicing. Circulating fibronectin is produced primarily in the liver, and this soluble form of fibronectin is referred to as plasma fibronectin. Cellular fibronectin is produced in a variety of different tissues by resident cells, including connective tissues, bone, and periodontal tissues. Antibodies have been generated that distinguish between plasma and cellular fibronectin based on sequence-specific epitopes that represent the individual forms of fibronectin. Similarly, the splice variants of fibronectin have been shown to exhibit differential solubility and sensitivity to protease digestion, suggesting that expression of each fibronectin variant can influence the fibronectin network formation and also its stability. Further, like the laminin, fibronectin assemblies also into a network on the cell surface. Fibronectin are basically very important and crucial for angiogenesis. Further, a glycoprotein dimer, which is connected by disulfide bonds at one end, can exist in soluble or fibrillar forms. Further, fibronectin is binding integrins linked to the cytoskeleton through scaffolding proteins that allows forces generated by the actin and myosin cytoskeletal elements to pull on the fibronectin to expose an assembly site that is not available in the folded or soluble conformation. The tension generated by the actin cytoskeleton transferred through integrin engagement reveals the site that initiates the fibronectin polymerization. Further, the force that is required for fibronectin fibrillogenesis, synticans, the transmembrane proteoglycans, they are responsible to enhance the fibronectin integrin engagement on cell surfaces and also influence fibronectin assembly. Once fibronectin fibrillogenesis is initiated, a series of steps that are not completely understood convert the polymerized fibronectin into an insoluble ECM. Formation of insoluble fibronectin likely involves protein interactions and additional conformational changes that result in a stabilized extracellular matrix. Notably, several experiments have contributed to the current model of fibronectin network formation that were predominantly carried out in two-dimensional cell culture systems. So this is a brief overview about the fibronectin and its synthesis. So basically, there is co-alignment of extracellular fibronectin fibrils and intracellular actin filament bundles. In this image, the red shows the fibronectin fibrils and green shows the actin filament bundles. Not only these are important for adhesion, but also they are important and vital for the migration of the cells. So it's not that the cells are attached in a matrix, but it is also important that the cells can communicate between each other and required signaling molecules are transferred and transfused, and also it allows for the cell to move. Laminins are the high molecular weight proteins of the extracellular matrix, which consists of three polypeptides, which is the alpha chain, beta chain, and gamma chain. So they are a major component of the basal lamina, which is one of the layers of the basement membrane, a protein network foundation for most cells and also the organs. The laminins are an important and biologically active part of the basal lamina, which influences the cell differentiation, migration, and adhesion. Laminins are heterotymeric proteins that contain an alpha chain, a beta chain, and a gamma chain, which is found in 5, 4, and 3 genetic variants respectively. The laminin molecules are named according to their chain composition, and thus laminin 511 contains alpha 5, beta 1, and gamma 1 chain. So in this way they are nomenclaturally named in order of their alpha, beta, and gamma chains. 14 other chain combinations have been identified in vivo conditions. The trimeric proteins tend to intersect to form a cross-like structure that can bind to other cell membrane and an extracellular matrix molecules. The three shorter arms are particularly good at binding to other laminin molecules, which allows them to form sheaths. The long arm is capable of binding to cells, which helps anchor the organized tissue cells to the membrane. The laminin family of glycoproteins are an integral part of the structural scaffolding in almost every tissue of an organism. They are secreted and incorporated into cell-associated extracellular matrices. And further, laminin is also vital for the maintenance and survival of the tissues. Defective laminins can cause muscles to form improperly, leading to a form of muscular dystrophy and also lethal skin blistering disease, which is known as junctional epidermolysis bullosa, and also certain defects of the kidney filter, which is known as the nephrotic syndrome. Basically, three kinds of basal laminae are there, which is a thin and flexible mass that underlies all epithelial cells and tubes, and not only for structural support and filtering, but also to determine the cellular functions. As it is seen in the image, that the basal lamina, it consists of the muscle cell plasma membrane and the connective tissue. Further, in the epithelial, the lumen or the external surface, and also it is found in the kidney glomerulus. So basically what happens is that the basal lamina are extracellular structures that are found closely opposed to the plasma membrane on the basal surface of epithelial and also the endothelial cells, and they surround the muscle and the fat tissues. While the primary function of basal lamina in most adult tissues is probably supportive, but significant evidence indicates that during embryonic development, basal lamina may be involved in regulating the heterotypic tissue interactions. Changes in composition of the basal lamina of salivary and mammary gland epithelial tissues during development probably are important for branching morphogenesis, which leads to the final form of the organ. Going into some details, so during tooth organogenesis, selective basal lamina degradation and direct cell contact between developing epithelium and mesenchyme has been documented and suggested to be necessary for cytodifferentiation. Basal lamina turnover, as suggested by these morphological observations, no doubt involves both basal lamina synthesis and degradation. We have studied several factors evidently required for basal lamina reconstitution in vitro conditions, and also have discovered that fibronectin added to the enamel organ epithelial cultures provided cues required for basal lamina formation in vitro conditions. So this is some basic information about the basal lamina. Further, if you want, I can make more videos related to more details of this or glycoproteinous structures. So electron microscopy again shows that the basement membrane consists of three layers, which is the lamina lucida, lamina densa, and lamina fibronetriculus. So the basal lamina is basically formed by the glycoproteins, the adhesion molecules such as laminin, fibronectin, entactin, fibrin, and thrombospondin. Various proteoglycans such as heparin sulfate that can influence viscoelasticity, permeability, homeostasis, and thrombosis, and microfibrils of collagen 4 and fibrin are secreted by them. Basal lamina borders the endothelial cells on their abluminal surfaces, and they form generally continuous boundary between the endothelium and the immediately underlying intimal structure. It is basically involved in the regeneration and attachment of the endothelial layer, the blood vessel permeability, initiation of blood clotting, and also provides a barrier to cellular migration. It connects the endothelial cell compartment to subjacent cell layers and the smooth muscle cells. The basal lamina consists of two zones, a clear inner zone which is known as lamina rara and a dense fibrillar zone which is known as the lamina densa. And both of these are adjacent to the interstitial connective tissue. The clearer inner layer contains the glycoprotein laminin, whereas the dense layer is composed of type 4 collagen. The basal lamina provides a pliable extensive bond which permits the endothelial cells to comply with changes in configuration related to cardiac pulsation and to vessel torsion and flexion. Furthermore, discrete adhesion points of increased electron density have been observed between basal peripheral cytoplasmic dense bodies of endothelial cells, which prevents the slippage of the lining cells when shear stress increases. As stated, that the basal lamina is formed by the glycoproteins, the adhesion molecules such as laminin, fibronectin, intactin, and thrombospondin, various proteoglycans such as heparin sulfate can influence viscoelasticity, permeability, homeostasis, and thrombosis. And microfibrils of collagen type 4 and 5 are secreted by the endothelium. Mechanically, the basal lamina, they tend to strengthen the vascular wall. Collagen type 4 chains form a covalently stabilized framework and provide mechanical resistance. Laminin forms a second polymer network and connects endothelial cells as well as heparin sulfate to the collagen. Intactin binds laminin and collagen, and fibronectin transmits the mechanical forces between integrin receptors on smooth muscle cells and the extracellular matrix to redistribute the mechanical load within the arterial wall. Thus, the monolayer of the endothelium is normally quiescent with cell-to-cell contact, inhibiting the proliferation, resulting in a low mitotic index, contributing to the stability and integrity of the vessel wall. In this state, the endothelial cells form cell-to-cell junctions, synthesizes and secretes extracellular matrix and basement membrane components, which includes the fibronectins, laminins, proteoglycans, and collagen. The underlying reticular layer of the endothelium is composed of collagen type 1 and 3, which are produced by both endothelial cells and smooth muscle cells and are the most abundant of the interstitial collagen. Certain evidence they suggest that the ECM components influence cell phenotypes in endothelial cells and smooth muscle cells. It has been certainly found that in culture, the endothelial cells phenotype is influenced by growth media, surface, and the age of the cultures. Further, cultures of the cells on various surfaces including collagens, fibronectins, and reconstructed basement membranes demonstrate different rates of production of gap junctions, actin, messenger RNA, which is also known as mRNA, protein, cell proliferation, and membrane specialization. There are several functions of the basal lamina. So the basal lamina, it provides support to the overlying epithelium and it limits the contact between epithelial cells and other cell types in the tissue and acts as a filter, allowing only water and small molecules to pass through. So basically, these in neuromuscular junctions, besides supporting or filtering the functions, provide the support. It is found in the muscle cells, which is known as laminin, and neuronal cells, argin, which is the heparan sulfate proteoglycan. So, the basal lamina is basically made and maintained by the cells that sit on it. It acts as a point of attachment for cells. However, it can also have other functions such as a permeability barrier in the glomerulus, which is to produce the urine. So with this, I end my today's class, but this session or this topic is not over yet, as I am giving detailed lectures on it. So there will be more 2 lectures as part 2 and part 3. So I hope it was helpful for you people who wanted to know about this. And please don't forget to like my videos and subscribe it to my channel, and also suggest it to your friends. So with this, I end my today's class, and I'll see you in my next class. Thank you.